Good afternoon, everyone, and being all together over this Memorial Day holiday. So in this session here, we'll have Sue come up on stage and join us on the panel here, if you can come and sit next to Barry. So I have the pleasure of being joined by Barry Burns, professor and associate chair at the University of Florida, Powell's Gene Therapy Center, and Susan Ward, the founder and executive director of CTAP, and she'll tell you a little bit more about this organization and what her team has been doing regarding data. So over the next 20 years from Peter Stein, that we have one shot on goal when it comes to study design, and so it is very critical for us to get this right. So what we want to do is go through some lessons learned and implications for the future as we do clinical trial design. I'm Lauren Morgenroth. I'm the CEO of TRINS. We are a neuromuscular specific and Duchenne focused CRO that was formed based on the unmet need for having a dedicated neuromuscular service provider. So it was you all in the room that said about 10 years ago you wanted to have a neuromuscular specific CRO, and so we partnered with the patient. These are the themes that we're going to be covering in today's panel. So we'll go through a little bit more about the need for drug development, what the drug developers need to make a clinical trial design, what have been some of the failures and why, minimizing the use of placebo and talking about maybe some controversial themes on that, talking about why certain trials are designed to be longer than others, and really thinking about who are all the key players that should be participating in the trial design. So with that, I'll hand it over to Susan.
Big green one, easy to see. So CTAP is a nonprofit scientific organization that we put together almost 10 years ago. I'm particularly grateful to Cure Duchenne, Mike Kelly, because he helped my partner in crime, James Signorovich, and myself put CTAP together, and Deborah, who gave us seed funding to start. A clinical trial is supposed to answer clearly and definitively one question. Well, maybe two, but one question: does this drug work? At the time when we put CTAP together, the community was facing really a pile of bad news. These were some of the largest trials that had been conducted at that time, and not one of them showed significantly, statistically significant clinical efficacy. Moreover, there were another 4 compounds that were already in clinical trials that everybody worried were going to suffer the same fate. So maybe all these drugs were duds, but maybe the trials were not good enough to be able to detect whether or not each of those drugs was in fact efficacious. And we have a clue that that might be the case in that far right column, which shows the variance that was seen in these trials. And essentially, that's the amount of noise in the trial. And the bigger that number is, the less happy we're going to be. Those numbers are bigger than they were expected to be in most of these trials. And they were too big for the trial to be even possible to detect efficacy at the level that was expected. So what we hope by the time that we're finished here today is that you will understand how a clinical trial is designed and why designing a trial in Duchenne is especially difficult. I think you'll see that we've made a lot of progress, though, and when we talk as a group, we will help you understand what you might expect to come next. So designing a trial is really like a balancing act. All trials principally along the following lines. In a disease like Duchenne, where you have declining functional ability, you can see that blue line and the blue dots there represent placebo, where you've got decline over time, over the duration of a trial. The patients who are treated with the therapy, one hopes, have a different outcome in the trial, and that they will decline less, possibly even gain function. Now, the key here is that treatment effect is the difference between the green and the blue lines at the end of the trial. And the key is that you have to be statistically significant to be able to say that your drug works. And if you see those error bars, which I know you've seen on many of the slides over the last couple of days and today, when they overlap, essentially you do not have statistical significance. So the name of the game here is to figure out how you can get some white space between the blue and its error bar and the green and its error bar. So how can we do that? There are several ways we could try. One way is to test a higher dose. A higher dose might give a bigger treatment effect, increase the space between the blue and the green. However, in Duchenne, most of the trials are using the highest dose that is safe enough to test. So we really don't have any room to take this approach in Duchenne. The most common way that this kind of problem, by the way, this kind of problem is common in all diseases, but the most common way in which this kind of problem is overcome is to run a bigger trial. And if you run a bigger trial, you end up with smaller error bars, and you stand a better chance of seeing a separation between the placebo and the treated groups. Well, this is a rare disease. Running bigger trials is not what we want to be doing. So a third way of achieving significance is to run a trial for a longer period of time. And if you imagine extrapolating those lines out, you can see that if you ran the trial for longer, it's likely that you would, at some point, see that separation. Now, if designing a trial is a balancing act in the first place, when it comes to designing a trial for Duchenne, it's really a high-wire balancing act. And the reason for that is, as you all know, boys grow in strength over time as they grow older. There comes a period of time when their ambulatory function is kind of stable, it's not improving anymore, and then eventually it declines. So this is not a simple one-line decline that we're dealing with. And the consequences of that are that your decline in the placebo arm, if you've got a mixture of patients in your trial, you're going to have, on average, a smaller decline in the placebo arm and a bigger error bar because you've got a mix of different types of patients. Some are improving, some are stable, some are declining. So in fact, it's even harder in Duchenne than it is in many disorders. So we have built predictive models to try and help drug developers overcome this problem. What you can see by these colors that show different phases of the disease is that the models that we build take into account all of these phases. They're not looking just at decline, they look at all of those phases. And on the right, complicating matters further, you can see that individual patients are in those different phases at different ages. So it's not that everybody progresses at the same rate. Patients progress at different rates, and that complicates the error issue even more. So here's an example of a predictive prognostic model. And what you can see is that the little graphic on the right there, you see that the individual spots line up rather better than the ones on the left. And what these axes are is predicting a one-year change in function versus what actually happened in given patients. Now, you can see that that prediction is better, and it's actually twofold better than the traditional or conventional way that predictions have been made in Duchenne. And that means that the unexplained variance, and I apologize for the technical term, is reduced, it's halved, more than halved. And the consequences of that are that the error bars get smaller, so you have a higher probability of being able to detect efficacy if the drug, in fact, works. The other thing that this can do for you is to reduce the size of the trial so that fewer patients are receiving placebo. So we can reduce the error bars without having to recruit a lot more patients. Now, we can build those kinds of predictive models over a longer period of time as well. And this slide just shows that we can predict time to loss of ambulation on average, and we can discern patients who are close to that event versus ones who are much, much further away. This use of loss of ambulation may not be the best time to event clinical outcome to use in a clinical trial, but it's really, really important when you're talking to payers who want to see evidence that your drug is working, not just in the clinical trial, but over time. So I'm going to skip that. I won't skip that. I will go back. So you may wonder why we pick the kinds of inclusion and exclusion criteria that companies do. So the inclusion criteria is all about ensuring that the patient who is going to be enrolled in the trial is sufficiently ambulant, sufficiently strong, that they can be tested using the NSAA. The exclusion criteria are an attempt to say, maybe we can enrich the population of patients for those who are at a certain phase of their disease progression. One way of doing that is shown here schematically with rise from floor. So children who are getting stronger, whose NSAA scores are still going up, they tend to have very fast times to rise from the floor. Over time, they get slower to rise from the floor until eventually that ability is lost. And as that lengthens out, patients are more likely to be in the stable or declining parts of the NSAA curve. So if you see a trial that is asking for patients to be excluded if they have a time on a time test that's, in this case, rise from floor greater than 5 seconds, then what that tells you is the company is trying to maximize their ability to see efficacy by focusing on patients who are more similar to each other at baseline. This one I think I'll leave because it's a bit more complicated. Before we move to the panel discussion here, Deborah wanted me to emphasize something that is really important to note. So, and that is the length of a clinical trial. This is one of the trials on vamorolone, and like other steroids, it's very clear that you can see an effect within 6 months. That last data point is 6 months, and patients get stronger when they're on a steroid, as you all know. However, not all compounds are steroids. So this is a graphic of Ataluren, which, as you know, has conditional approval in Europe. And one of the requirements for that drug is that they need to follow what happens to patients who are on the drug and compare them to patients who are not on the drug. Now, we know from earlier that Ataluren did not show efficacy at 12 months in 2, in fact 3 separate clinical trials. They did not achieve statistical efficacy. However, this graphic is over a 5-year period of time, and what it shows is patients who are losing ambulation. And the median time for that, for patients not on Ataluren, was 13 years, roughly, 13 years of age, whereas patients treated with Ataluren, the median time to lose ambulation was 17 years. So clearly, we have efficacy here. Similarly, with Etal Pharmaco's compound Givinostat, they chose to run a trial that lasted 18 months. This was atypical at the time, and you can see that they have statistical significance there. But that arrow, the arrow that I've laid on top of this graphic, shows that had they run this trial for 12 months, they would not have seen efficacy. So that made those 6 months made the difference between being able to submit to regulators and say this drug works versus not. So that's it for slides. It's late in the day. We don't need any more slides. So I'm handing back to Lauren.
Thank you, Sue. So with that, we'll have about 5-6 minutes for open up for discussion. Let me just skip through these. They're just a placeholder for us to skip ahead. Let me go back. So I'll leave this up and use the opportunity to start off the panel discussion with the topic of eligibility. And so I think many families in the room are faced with finding lists of eligibility, inclusion, and exclusion criteria that often scroll pages. And so I want to just open up for discussion on why are there so many inclusion and exclusion criteria. So maybe Barry, if you want to start.
Yeah, thanks, Lord. I can comment that, a sponsor that knows a certain mechanism of action of their product wants to identify patients who are going to have a measurable outcome that is influenced by that drug. And so this may mean, at least across the spectrum of outcomes that are considered in Duchenne, obviously we're all very focused on skeletal muscle function. So, and there's been a lot of, Susan showed that natural history data that shows that as boys gain skills, they reach this then plateau period, after which they start to lose skills. And so the drug developer may realize that in order to identify a change from their starting point to the maximum effect of the drug, they have to consider how long a period that would be and at what point a child is starting into a study to see where they might end up. And sometimes this is referred to as ceilings and floors in the effect. If an individual is close to the unaffected functional measurement, they can't really improve from that. They can only decline, and that's not what the drug is intended to show. And if they have too low function at baseline, they may not respond to the mechanism that's being studied. So people are chosen then between those two functional measurements. That is what ultimately gets decided on as the primary outcome measure of the trial. And the trial will survive, succeed or fail based on that outcome. And sometimes there have been considerations to do composite endpoints where there are many different metrics that are used, but then any one of those that doesn't meet the goal of the outcome drags down the others. So it's a very challenging aspect of study design to choose those right endpoints related to the drug that's being studied. And you as participants in these studies and parents of participants get a very fast lesson on what makes sense to you. And it really, I'll say, even though there's a lot of experts may agree or disagree on certain things, you as the participant have to understand it well enough to say, I get it, and this makes sense, and my son could cooperate with this study. He will do his best and be a valuable participant in the study, and that benefits him as the participant and also the sponsor in designing the best study. So I'd say that's what goes into it, and it's somewhat art as well as science.
So for the sake of,
I knew that was going to happen. Sorry about that.
so for the sake of time, I'll kind of take one other topic we really wanted to get through is this whole concept of minimizing bias, minimizing placebos, and so I wanted to open it up and see and pose something that I've heard families tell me over the last 2 days is it actually ethical to have a placebo in a design that's in a non-ambulant or an adult patient population? So,
Susan, you want to comment?
One of the challenges of using natural history, which it would be an obvious thing to do if you want to compare a drug to non-treated, would be to use a natural history. But one of the concerns is that if you are in a clinical trial, these tests, NSAA, the time tests, they all are subject to effort. So, you know, if the boy is really tired today, or if he's really enthusiastic, the time that it takes to conduct the test, or even the score on the NSAA, may vary from one day to another. And so the concern is that a placebo arm is in the trial, so anything that is motivational around a trial is the same, quote, for treated and for placebo, but that may not be true for natural history patients. So there's a concern that there might be bias. And one of the things that we in the field are doing is working very hard to understand how big that bias is and to account for it so that we can be in a position where we can convince regulators that we know how to handle the differences, if there are any, between placebo and natural history. Elaborate.
Yeah, so I'll add one thought to this, is that pediatric studies actually that are beyond minimal risk have to be designed in a way that provides an opportunity for direct benefit. This is part of the federal regulations regarding clinical research. So it's really only in adult populations that you'll see a study in which subset of patients who can be informed about the risks and benefits would never receive the therapy. And this is almost never done in a pediatric study. There was one instance where there was an early indication that the untreated population actually had higher mortality, and that caused the study to be terminated really almost immediately. So now I think we see in the Duchenne space that there is recognition of that need for direct benefit in the patient population, but there is a delay between, and almost all studies now are not really truly placebo controlled. They're a delayed treatment design in which a subset, whether it's a third or a half of the patients, will get the product later. And really becomes a question of then how much later and what happens in that interval. And one of the things that's determined in that delayed access to the treatment is you can assess, the sponsor can assess the inherent safety events that are related to the disease. So boys that are transitioning to wheelchair use might be subject to falls and fractures, for example. And how would you determine whether that's happening randomly among people who don't get a treatment versus ones who the treatment might predispose towards that? So there's usually a delay addresses safety considerations or laboratory abnormalities that are changed by the drug. But those safety events are generally, at least in the biologics that are being used in Duchenne treatment, are generally right around the time of the treatment within the first few months. They're not 6 months or 12 months later. So I would argue that if the sponsor's interested in differentiating safety events between the treated and delayed treatment groups, that the period of delay could be dramatically shortened. And it really doesn't benefit, particularly as I think to your point about a non-ambulant population that is not improving, they're only losing function, that a prolonged delay is not advisable. And I think ultimately it's up to sponsors to have that negotiation with the FDA because these are not large studies. There's not really a benefit to the study design to have a large cohort of untreated patients that lose function to the point that they're not really comparable to the treated counterparts that were treated earlier. So that's just been my bias, at least in Duchenne, the non-ambulant population, that we should consider novel treatment designs that eliminate the delay or minimize it. And I'll go one step further to say that we now have a technology that allows, you often hear about gene therapy studies being once in a lifetime, and that is not entirely true. We're conducting a study now in another muscle disease where we randomize patients to 2 different dosing groups. And the intent is to not to assess the safety differences between 2 dosing groups, but to be sure that all the participants, even at the time of enrollment, receive the most efficacious dose. And so randomized dose escalation study design is one that's not been used before in the gene therapy arena. The most effective one is where you change the dose and then change it back again. That's something we can't do, unfortunately, with the gene therapy product, because once it reaches all the cells in the body, it never leaves there. But we can cross over people between low dose and high dose, or effective dose and a more effective dose if that's possible. And that also gets to the point of whether there's a benefit to incremental dosing to get to a certain desired effect. Dose titration, which is another way small molecule drugs are studied. It's called a multiple ascending dose study design, in which patients or study participants receive increasing amounts of a drug to identify safety events or the maximal efficacy. So I think we may ultimately get to that point even in Duchenne gene therapy studies, because not everyone, as you can see in the study results you've seen over the last 2 days, not everyone is the same. And some boys may respond very favorably, and others may need a greater level of exposure. So that's something that's coming, I hope, in the Duchenne space.
And I think we're definitely seeing more creative study designs where the whole ecosystem that's planning those clinical trials is really thinking about all those important items of when should there be a crossover, is the crossover applicable, and what's that time frame that you're essentially delaying that possibility of being on the interventional. So we are over time, but I did want to close out by saying study design is something that needs to be done collectively with the families, with the academic centers, with the sites to understand the barriers to being able to operationalize the clinical trials. Obviously, the regulators need to play a critical role, and that was shared with us with the FDA. And so this is something we want to have everybody at the table to be able to make sure that that one trial design that will be used for approval is done right. So thank you, everyone.
Thank you.
And I will hand it over to Paul to give us the updates. Paul, are you coming up to give us the updates on the breaks and other fun activities?
He's the chief fun officer, so get on up here.
Fun foodie.
I got this. I'm good. I don't need that. All right. Thank you. Great session. All right. Here we go. Last break. We got 15 minutes. Get back here for a session, and then don't forget tonight, Ohana, a little Oaha. It's going to be fun, and I know it's probably a little bit long of a day. Keep going. We're almost there. So thank you.